yes, as you just heard, I will be talking about NMR spectroscopy of some heterogeneous catalysts. And these catalysts are not on this slide, they're on this slide. Right, these catalysts are relevant for the production of acrylics. And this is quite a group of versatile and stable polymers that have a range of beneficial properties that allow for them to be used in an equally broad range of areas. So just to give a few examples, they are biocompatible, so they can be used, for example, in bone cements, and they possess high strength and optical clarity, so they can be used in acrylic glass, which is something we encounter in our day-to-day lives. The annual global production of polymethyl methacrylate, or short PMMA alone, which is one widely used acrylic polymer, is about 4 million tons. And that large number can, or maybe should, motivate us to think about how we can improve the ecological footprint of this industrial process. So one potential roadway would be to think about recycling PMMA, which is research that is being undergone, but in this project, we were thinking more about the production process itself. So PMMA is made from the methyl methacrylate, or MMA monomer. And if we go one step further back, again, the MMA monomer has to come from somewhere. And one potential route to make it is the so-called alpha process, where formaldehyde and methylpropionate undergo an aldol condensation to give MMA and water. And as you can see here, this is done over a catalyst based on amorphous silica. And we can view this catalyst as a sort of key in order to think about how we can improve this process by reducing waste byproducts and energy consumptions through improvement of the effectiveness of the catalyst itself. So how do we do this? Well, first of all, we have to understand the relation between the composition and the catalytic activity of this material. So if we go in the lab and we make a material which is comprised of amorphous silica, as I said, and then zirconium and cesium deposited onto it, and we choose a certain surface loading, how will this affect the activity? And we can measure these relationships empirically and learn something about, for example, how the concentration affects the activity. But in order to really tune this catalyst and improve its effectiveness systematically, we have to understand how the structure informs this relationship. Now, based on what I told you about this material, you probably already guessed that the structure is quite disordered. It starts with the amorphous bulk, and then you deposit two different things on the surface. So there will be a range of structural motifs and probably no long-range order at all. But from an NMR perspective, this can be quite exciting because there's quite a lot of NMR active nuclei in this material that we can use as probes into the surface of the structure and not think or worry about the long-range order too much. So this is what we underwent, and in the interest of time, I will only be showing results from cesium and oxygen NMR spectroscopy of these materials. And we did this following two different approaches. We had a library of model compositions where the composition was systematically varied and properties were determined. And we also synthesized some crystalline model components where we can look more in depth at the relationship between the structure and the NMR parameters. So let's start by looking at how cesium NMR can tell us more about the local structure of the active species on the surface of this material. Cesium-133 is a quadrupolar nucleus, but its quadrupolar moment is relatively small. So compared to, for example, sodium-23, line widths are quite narrow. For example, for some clay minerals containing cesium or cesium salts, we expect line widths up to about 25 ppm. But when we initially started this project, the line widths that we measured for the catalyst materials were significantly larger than that, 2 to 3 times to be a bit more precise. So the first question that we were concerned with was what the origin of this broad line is. And we can answer this by varying the magnetic field, including going to high magnetic fields to see how the line width changes. And what we see here in this comparison is that the line width is in fact not significantly reduced if we go to high fields. So we can understand that this broad line is the result of chemical disorder of the active species, even if the surface loading is extremely low. So what do the chemical shifts that we observe mean? What can this tell us about the structure? In order to attempt to better understand this, we synthesized two crystalline model compounds, two cesium zirconosilicates, where we were hoping that the local environments of cesium with ZRO and SiO linkages in the vicinity could maybe mimic the behavior of the active species on the surface of the catalyst. And you can see here that the NMR spectra that result from these materials are quite different in appearance. They actually exhibit well-defined and narrow signals. So that's quite a stark difference, but it allows us to look at the relationship between NMR parameters and structure. Without going into great detail of this large table, I put this here to show or to illustrate that we looked at various structural parameters, and that overall there's unfortunately not a simple trend that allows us to just say X parameter changes, chemical shift goes up or something like this. But maybe as an overall trend, it seems that larger chemical shifts seem to be associated with shorter bond lengths and larger coordination numbers, maybe depending on how exactly we define those. And maybe unsurprisingly, the quadrupolar coupling constant is related to the distortion of the local structure. So we could try and extrapolate this now to the catalyst spectra. But there's a fundamental difficulty here, and that is that none of these materials exhibit negative chemical shifts. So it's possible that some of the cesium species on the surface of the silica exhibit local environments that are somewhat similar to what we see here, but a large portion of the material actually does not. And something else that we haven't touched upon so far is that these line shapes that we see here, at least at high cesium loadings, don't seem to be the result of a unimodal distribution of parameters. For the, maybe I'll go back, for the light red line, there's clearly a superposition of at least two components at play here. So given that everything is more complicated than originally anticipated, we started controlling the surface, the state of the samples more rigidly. First of all, by removing surface water from the material under vacuum and at elevated temperatures. And as shown here, by comparing the dry samples and dry spectra of dry samples in shades of red with as prepared samples in shades of blue, removing the surface water completely rearranges the local cesium environments of the surface. This is illustrated by more negative chemical shifts, the removal of this narrow spectral component, and an overall shift of the signal maximum. We can explain this if we assume that removing surface water results in more covalent SiOCs environments to be formed. What we also see is that by comparing samples that don't contain zirconium or whether zirconium has been introduced by the means of different precursors, is that the difference between these compositions are by far exceeded by the difference between the dry and the hydrated sample state. So the surface really seems to be dominated by the presence or absence of water. To go in the opposite direction, we built this very sophisticated controlled humidity environment where we could subject samples to varying degrees of water in the atmosphere. And if we do this, and if we leave samples in there for longer, or if we increase the water concentration, going from shades of red to shades of blue, we can see a gradual and continuous increase of the relative intensity of this narrow component until it coalesces at around 0 ppm, resembling cesium in solution, even though the sample is still solid in this vial. So this led us to believe that the narrow component is associated to a more mobile cesium species. And we can further test this by looking at the relaxation behavior of the two components. Shown here are comparisons of spectra recorded at different recycle delays. And if we look at the spectrum in dark blue corresponding to the shortest recycle delay, we can tell that the narrow component relaxes more fast, and also that it doesn't contribute to the spinning sidebands, indicating a smaller CQ we can further confirm this with saturation recovery experiments. So altogether, these results tell us that we need to rethink how we think about the surface of this material, and that the differences that we see are not so much related to the different species that are present and bound to the surface, but more that there is a coexistence of a hydrated and a dry state in which the cesium can assume. The hydrated state we think of as a surface water film, where the cesium is mobile and isotropic, and then in the dry environment can bind to a variation of environments more covalently. And if we then check what the influence of cesium loading is in these two different environments, we can see that if we increase the cesium concentration, the narrow component of the spectra grows in, indicating that at some point, rather than binding to the surface covalently, additional cesium species agglomerate in this surface water environment. So having learned more about the local structure of the active species, we turn to oxygen NMR spectroscopy to see whether this can tell us more about the connectivity on the surface, not just with the cesium, but between cesium, zirconium, and silicon. But in order to carry out oxygen NMR, we first have to artificially introduce the oxygen-17 isotope, which is NMR active, into the surface, which leads to enormous time savings in the NMR experiment. There's a variety of methods that we can choose from to introduce this isotope, either during synthesis or after synthesis. And all of them rely on having the sample be exposed either to isotopically enriched water or isotopically enriched oxygen gas. In the case of a catalyst material, where there's a difference between the bulk and the surface, and especially having just seen how the surface interacts with anything we put on there, we do have to be very mindful about how the enrichment changes the surface, though. For example, maybe the most gentle method out of all of these that I've just shown that we could pick is the so-called wetness impregnation, where we take a rota's worth of material and we drop a couple of drops of isotopically enriched water on the surface. We let it sit for a couple days or weeks, and then we dry it and we record an NMR spectrum. And just to illustrate, if we do this for the support material and record a 3QMS spectrum, we can resolve the difference between the xylanol and the siloxane species on the surface. Can we do the same for the catalyst? Yes, we can. So repeating the experiment for a material that has zirconium and cesium on surface results in a similar picture. And just to stop here and pause for a moment, this is quite interesting because it means that we can introduce this oxygen-17 from the water into the surface at ambient condition. And the water doesn't just change the environment of the cesium, but also interacts with the silica surface itself. If we now stick with this method and compare the influence of different compositions, and we do this at high fields to benefit from the improved resolution in 3QMS experiments, we can see that changing the composition results in differences in the line position and line shape, and also relative intensity of the two signals that we obtain. This is also illustrated if we look at spin echo experiments. And here we can see maybe even a bit more clearly than in the 2D visualizations that for the sample with high zirconium loading, the line shape changes, indicating that there's maybe a different CQ present or a different parameter distribution. And we also see that we have to think about how the composition affects the efficiency of this enrichment. So how efficient is the enrichment, or what happens if we move away from this gentle method of just dropping water on the surface? Well, here's a comparison of different isotopic enrichment methods that we tried. Two of them are, again, the incipient wetness. Then we ball milled the sample with water. We steamed it in an autoclave, and we went to do gas enrichment, which is maybe the classical method of enriching oxide materials. All of these methods result in different intensities and different line shapes, but this comparison isn't quite fair because we do have to take into account how much water we are actually using. And as you can see here, this makes the ball milling suddenly look a lot better. We can't quite compare the gas enrichment, but we can acknowledge that one gas enrichment experiment is significantly more expensive than one water-based experiment. So overall, it seems that hydrothermal steaming in this case was the most efficient at introducing oxygen-17, but it is a water-based method, and we know this will affect the local environment of the cesium. And we could circumvent this by using gas enrichment and not introducing water. But as you can see in the schematic here, this method involves heating the sample up to at least 600 degrees. So that could also lead to surface degradation in some other form. All of these are still questions that we need to continue answering going forward. And with the remainder of the time, I wanted to touch upon one last thing that I haven't mentioned yet. And that is that so far I've only really talked about silanol and siloxanes, but the catalyst does have zirconium and cesium on the surface. So where are the ZRO and the CSO linkages? Based on literature review and DFT calculations, we can estimate where we would expect to see them. And if you are like beneficial to these experiments or like open-minded, you might see, okay, maybe there's a bump in the baseline that could be a signal, but it could also just be a bump in the baseline. So what's going on? Do we not see it because there's not much of it? Or did we pick methods that are more susceptible to enriching SiO(Si) bonds, and we don't enrich these species as efficiently? We can answer this by enriching a sample with a significantly more high zirconium content. In fact, one that would be not useful as a catalyst, but is useful as a model. And in this case, we do see a signal that is in the right spectral region and that we can attribute to SiO(ZR) bonds. And I want to highlight that the blue line here, again, is the incipient wetness impregnation. So it's not just the SiO(Si) bonds, but the SiO(ZR) bonds as well that open at ambient conditions and exchange with the surface water. Finally, we can go back to the model materials that we used earlier to look at cesium NMR parameters and see what kind of oxygen environments are present in these zirconosilicates. And if we look at the predicted chemical shifts, it's obvious that there are two different regions in which we would expect chemical shifts to appear that we can attribute to SiO(ZR) and SiO(Si) species. And in both cases, whether or not cesium is present or not only has a secondary influence on the apparent chemical shift. If we predict NMR line shapes for the two materials, which are shown in black and gray, compare this to experimental spectra of catalyst materials, which are shown in red. First of all, what we see here is that one of the SiO(Si) signals agrees quite well with our experimental data, and that begs the question as to whether the SiOCs species, in this case as well, only have a secondary influence and are somehow hidden underneath this broad line shape. And secondly, if we add zirconium onto the material, we do get the signal that I just showed, and it's in a similar spectral range, but the local environment of the SiO(ZR) species is clearly quite different than in the model materials and also probably more disordered. So to sum everything up, I hope that I have showed you how we used cesium and oxygen NMR to probe the surface of this material, which is used for acrylics production, and that we saw that the local structure of the active species is highly disordered, no matter what the surface loading is, and that we have to think of the surface as containing two different species, one more covalently bound and one hydrated. And that in both cases, we saw that the surface structure is strongly influenced by the presence of water. And that it's not just the cesium that is in these hydrated or non-hydrated environments, but that SiO and ZRO bonds also open at mild conditions. So going forward, we have to think about how the local structure of this active site plays a role in the catalytic mechanism, and what is the role of these mobile isotropic species. With that, I'm at the end of my talk. I'd like to thank everyone who's listed on this slide, who contributed to the work, and of course also the Highfield facility for the experiments we did here, and to all of you for your kind attention. Thank you.